Hello, my name is Gholipour. I'm an associate professor in radiology at Boston Children's Hospital and Harvard Medical School. In this presentation, I will talk about diffusion-weighted imaging for the analysis of fetal brain microstructure and connectivity development. I have no disclosures. Diffusion-weighted MRI or DMRI has been the technique of choice for in vivo imaging of the brain microstructure. Through measuring free and restricted diffusion of water molecules and appropriate modeling, DMRI allows us to image the structure of the major axonal fiber bundles or fiber pathways that connect various cortical brain regions. There has been significant interest in imaging and studying the structure of the early developing brain in vivo or in utero using this powerful MRI modality. However, fetuses move significantly during DMRI acquisitions. Fetal motion often disrupts the spatial correspondence of the data that is acquired during the relatively long duration of DMRI scans. This would often lead to completely distorted images that are not useful. My goal in this presentation is to explain the research that we undertook in the past years to address the challenges inherent in fetal DMRI and show where we stand in terms of tools and resources that are available to study early development of the brain and the brain connectivity. Here in this image on the right, you see a color fractional anisotropy that we tried to get out of the scanner without any processing. And you see how distorted it is based on. This slide shows the outline of the talk. After an introduction, I will discuss our methods and results for slice localized motion tracking, robust diffusion tensor estimation, and then afterwards I will talk about our fetal DMRI atlas and fetal structural connectome analysis. In this slide, I have a plot that shows the course of the human brain development. The plot has been scaled in a way to highlight the period before birth. That means from conception to 40 weeks of gestational age, just before birth. And this shows in weeks, and the rest of it, as you can see, are in months and years. So what this plot shows is that a lot of the brain structure development happens before birth, during the fetal period of brain development. A fetal brain generates neurons at an astonishing rate of 500,000 neurons per minute during mid-gestation. That leads to about 100 billion neurons, and through the processes that are happening at this time, neuronal proliferation, migration, apoptosis, and synaptogenesis, about 100 trillion minimum number of connections are formed in the human brain just before birth. Of course, some of those processes continue after birth while the brain tries to build its functions. Fast forward, let me show you what is in the horizon for our journey into this work. The question is, what does fetal DMRI ultimately show us? Here are snapshots of the atlases that we built at 27 and 38 weeks based on DMRI images of normal fetuses that we scanned and processed with our techniques. Images on the left show axial, sagittal, and coronal slices of our fetal atlas at 27 weeks, which show many structures including limbic fiber bundles, corpus callosum internal capsule, cingulum, superior longitudinal fasciculus, forceps minor and major, all color-coded. We also see the radial coherence of the telencephalic wall that gradually disappears through the gestation as projection and association fiber pathways mature, and ultimately it forms a mature-like brain structure like the one that you see on the left, on the right. That is a 38-week fetal brain atlas. With that introduction, I will talk about our methods and results. As mentioned before, motion is the most important challenge in fetal MRI. So we start by addressing that. Our fastest DMRI scans take about 1 minute with about 3 seconds per volume. Fetuses move near continuously during the MRI acquisitions. So we ought to compensate motion not only at the volume level, but preferably at the slice level, as there is significant interslice motion in the MRI volumes. Plus, interslice motion may occur, and it may corrupt some of the slices, as you can see in some of these slices in this image. So we aim to capture motion dynamics through a combination of image registration and Kalman filtering for slice localization. So states here represent motion states for the T observations. Every observation is one slice acquisition at time point T, and we have these slices that are acquired from the fetal brain using the MRI. And so with the Kalman filtering or state space estimation, we model it as a dynamic process with those states and outputs with process and image noise. Our solution to it is nonlinear state estimation with Kalman filtering. So we calculate the similarity metric between the slice and the images acquired at time k and try to maximize that similarity. Due to registration errors, image noise is not normally distributed, so we opted to use an outlier robust Kalman filter. If retrospective motion correction is being done, then we can use the Kalman smoothing instead of Kalman filtering, which can be used with a window size of 5 time points for improved results. To validate and optimize the technique, we performed adult volunteer experiments where we attached motion tracking sensor to the forehead of the subject to measure motion, and the motion measured by that sensor has been shown in these plots on the bottom. As you can see, these are the motion trajectories for the 6 motion parameters, translations and rotations for that subject during that motion experiment. And if we do ordinary sequential slice to volume registration, these are the plots that you get with our approach. And with our extended approach, which is the robust Kalman filter slice level motion tracking, you can see that we eliminate those spikes and noises, and we get much better motion tracking as compared to the gold standard or the ground truth for this subject. Now that we had a way to estimate fetal motion with the technique that we validated, we wanted to reconstruct diffusion models. However, after slice level motion correction, data is scattered into this space. The problem cannot be easily solved through resampling on a regular lattice. We used a forward model and inverse problem approach, in which image data and gradient vectors were both adjusted and rotated to compensate for the motion at every slice. So in this approach, we end up with building a data structure with variable length diffusion measurements per voxel. On our target image, we rotated gradient vectors based on estimated motion and estimate tensors using weighted linear least squares. So this is the diffusion tensor modeling, and we solve it through minimizing this cost function here. The weights in our WLLS or weighted linear least square solution are important as they incorporate the slice profile and the distance of each slice voxel to the target volume in the MRI model that we have. In this formulation, we're solving for diffusion tensor model, and these weights take care of the scattered data interpolation and incorporation of slice profiles for us. So these are critical. First experiments, again in an adult volunteer who stayed still during one scan, which we use as gold standard for the brain fractional, color fractional anisotropy or color FA, and the subject moved in other scans. With a motion scan, if you just get the original data, this is what you get for the color FA. With the volume to volume registration, which is the current state of the art in the software packages, you will get an image like this. But if you use our motion tracking based slice to volume registration, you get a much nicer color FA that's a much better match to the gold standard. So the papers reporting these works were published in TMI, and the version for the fetal MRI was published in Neuroimage in 2017. Application to fetal MRI, again we're looking at color FA with different techniques: original volume to volume registration, slice to volume registration, and our Kalman filter based slice to volume registration, and with linear least squares estimation versus weighted linear least squares in the bottom. And we see much improved performance with our technique. This allowed us to do whole brain tractography with much improved results compared to other techniques, and this figure shows an in vivo DTI tractography for a 24-week fetus, which is quite impressive of how many connections and structures you see at that age. So based on that technology, we were able to scan a lot of fetuses and process images appropriately. We were able to build a population atlas, the first spatiotemporal diffusion tensor MRI atlas of the brain microstructure from in-utero images. And you see snapshots of these atlas. Basically, it was based on regression of images onto a manifold at ages with a kernel regression in age and diffeomorphic tensor-to-tensor registration between the tensor images. On this figure, however, I'm showing color fractional anisotropies. And now you see that, as I showed earlier, a lot of structures are present already in early ages of the fetal period. Our atlases are available online. It actually comes with a T2 atlas, which includes 120 plus parcellations of the brain regions, as well as the fetal diffusion tensor atlas that we showed. Regional tractography can be informative and can be performed on the images with the tools and atlases that we've developed. And these show how these corticospinal tracts and the other tracts are developing across age in the third trimester. And another snapshot of the corticospinal tracts, inferior fronto-occipital pathway and the inferior longitudinal fasciculus in the fetal brains. Now, with all of those technologies that we've developed, we made a processing pipeline that reconstructs a high-resolution T2 image of the T2 slices that are required of a fetus. With that, we can get atlas-based parcellations of the brain, and our diffusion MRI processing pipeline similarly corrects for motion, reconstructs images, does the tensor estimation, tractography in a robust manner, as we had discussed, and then that would lead to ROI-based connectivity analysis that can now show us the structural connectome of the fetal brain. These are shown for the atlases at 25 weeks and 35 weeks, and you see that a lot of information can be extracted from these images. This is unpublished work, and we're still looking into what exactly scientifically those connections and the graph theory analysis shows us. Just to briefly show, those analyses show that global efficiency and local efficiency both significantly improve over a period of gestation age from 23 weeks to 35 weeks. You see the points of the atlas, and small wordness property is observed across age for the fetal brain. In conclusion, we demonstrated an approach to process fetal DMRI acquisitions with slice level motion correction and robust model estimation. We also demonstrated the fetal MRI processing pipeline for fetal structural connectivity analysis using DMRI processing and atlas-based parcellations. We performed connectome analysis based on in vivo DMRI analysis, which showed a rapid increase in global integration and local segregation based on graph theory measures. And we also observed early lateral asymmetries in the development of major structural connections. The increase in connectome development complies with other studies in the recent years, such as this study by Altanat Heymurtash and using fMRI, which was published in several Cortex earlier this year. I would like to acknowledge my colleagues and contributors to this work, specifically those who directly contributed to DMRI processing, the pipelines, atlases, and its scientific value and applications have been listed here in blue color. And of course, there are other collaborators and colleagues who indirectly helped with a lot of material that led to this research. And of course, acknowledgement of funding from the NIH, Trasher Research Fund, McKnight Foundation, Fetal Health Foundation, Harvard Catalyst, NVIDIA, Office of Faculty Development at Boston Children's Hospital, and National Science Foundation.